Hello students, hi there. I welcome you all to this online class in which we shall take up this topic biosynthesis of sucrose and starch in plants. Now before going on to the topic, we'll take this opportunity to refresh your memories regarding the topics which we have already covered in this course before you guys proceeded on to this first vacation. As you can see, we have finished Unit 1, which included various topics ranging from plant cell organelles, their separation, structure, function, and photosynthesis. So it had around these 15 topics which we have already covered in the physical classes which we had earlier before this event happened. Now in this class I am starting Unit 2 of this course, which has all these topics: sucrose, starch, interconversion, biosynthesis of carbohydrates, proteins, lipids, nitrogen fixation, sulfate reduction, incorporation of sulfur in amino acids. Various topics are there. So in this unit we will be having these 5-6 topics, and among these 5-6 topics, around I think 6 topics, I'm going to take up this first one, which is biosynthesis of sucrose and starch in plants. So sucrose is a common disaccharide produced naturally by plants, and if you see, it is a carbohydrate, as we just mentioned. As I just mentioned, it is a disaccharide, so it's obviously a carbohydrate, and being a disaccharide, it is obvious that it will be made up of two monosaccharide subunits. So the two monosaccharide subunits which are present in sucrose are glucose and fructose. You can see the structure here. Both of them are joined together by a glycosidic bond, and this sucrose is extracted from many plants like sugarcane, principally from sugarcane, and even sugar beet also, and it is then refined industrially using bleaching processes, and finally obtained as a crystallized solid which is odorless, white in color, and it tastes very sweet, and it's commonly known as table sugar. It's a very common and important component of our daily life. It is used for preparation of several dishes, many other food products, and chemically, if you see, it is soluble in water, and the chemical formula for this molecule is C12H22O11. So it has 12 carbon atoms, 22 hydrogen atoms, and 11 oxygen atoms. Now, higher plants, or the green plants, you can say, they are characterized by their ability to use solar energy, which is further utilized to convert carbon dioxide into organic carbon in the process called as photosynthesis, which happens in leaves of the plant. Now, once this carbon dioxide is fixed in the leaves of the plant, they have to be translocated to different regions or parts of the plant. So leaf is regarded as the source. We have this concept of source and sinks relationship in plants system. So source are those organs or those plants of the parts of the plant which have the ability to synthesize organic carbon from carbon dioxide utilizing the process of photosynthesis. So leaf is a source, you can say, organ or source part, and it synthesizes carbohydrate using carbon dioxide in presence of light, which is the process of photosynthesis, as I just told you, And you have already seen in our previous unit that this photosynthesis has light reactions, dark reactions. The light reactions are dependent upon light, and the dark reactions are also called as Kelvin cycle or the C3 cycle. And the final product of that C3 cycle are three carbon compounds which are called as triose phosphates. Now these triose phosphates and all these process runs in the chloroplast of the plant cell. This is another thing which is important from the point of view of studying the sucrose biosynthesis, because you should know the site where sucrose biosynthesis happens. So if you see this diagram here, you'll see that sucrose biosynthesis is happening in the cytoplasm of the plant cells. And the starting compounds which are required for sucrose biosynthesis, or you can say precursors, they are the three carbon compounds, triose phosphates, which are synthesized during the Kelvin cycle or the C3 cycle, or maybe C4 cycle in case of sugarcane. So triose phosphates, once they are synthesized, so they will be synthesized, as you can see, in chloroplast. They have to be transported outside the chloroplast like this into the cytoplasm. Now, the chloroplast membrane will not be permeable to these triose phosphate molecules. So they pass through these chloroplast membrane to integral protein system, which is the inorganic phosphate translocator. So what this inorganic phosphate translocator does is that once the cytoplasm once the inorganic phosphate levels increase in the cytoplasm, it allows the diffusion of these inorganic phosphates into the chloroplast, and simultaneously it also allows the exit of the triose phosphate molecules into the cytoplasm. And later you will see that the concentration of these inorganic phosphates have a significant impact on regulation of sucrose biosynthesis. You'll see later in the slides. So what basically you have to remember here is that carbon dioxide, which is fixed in the chloroplast by photosynthesis, the final product, triose phosphate of the C3 cycle, is translocated outside the chloroplast into the cytoplasm through inorganic phosphate translocators, and once it reaches the cytoplasm, the process of sucrose biosynthesis starts, and it is dependent upon various factors. As I told you, one of that is the concentration of inorganic phosphates in the cytoplasm, and later we will see its role in the regulation of sucrose biosynthesis. So basically, sucrose is the end product of photosynthesis. It is a primary sugar produced and transported in the plants through phloem tissues. So that you have to remember, that once the carbon dioxide is fixed during the process of photosynthesis, many sugar molecules, you can say many organic carbon molecules will be formed, and sugar is the major molecule which is available in plants, and it is translocated or transported throughout the plant. So here is the simplified steps of sucrose synthesis in plants. Now you can see this is the structure we have already seen in the previous slide, and it is made up of this glucose molecule, which is a 6 carbon sugar, and even the fructose molecule, which is a 6 carbon sugar. Both are joined together by this glycosidic bond, and this is the chemical, you can say, IUPAC name of sucrose. It's like beta D fructofuranosyl alpha D-glucopyranoside. Now, if you see the steps, these are the basic simplified steps of sucrose biosynthesis. What you will see is that the UDP glucose, this molecule, it is activated form of glucose, you can say, in which UDP, uridine diphosphate, gets attached onto the first carbon atom, and fructose 6-phosphate are condensed in a reaction which is catalyzed by sucrose 6-phosphate synthase, leading to the formation of this molecule, sucrose 6-phosphate. So this is the first step. And this enzyme, sucrose 6-phosphate synthase, this is the principal enzyme for sucrose biosynthesis. And regulation of sucrose biosynthesis, as I told you earlier in the slides, is very much dependent upon regulating this enzyme. Many factors regulate these enzymes. So this enzyme plays a principal role or the major role in regulating the steps of for sucrose biosynthesis. So the first step is formation of sucrose 6-phosphate, which is catalyzed by this enzyme. Then further what happens is this sucrose 6-phosphate molecule is dephosphorylated and phosphate group is removed. This phosphate group you can see here. So this phosphate group is removed as inorganic phosphate, and ultimately sucrose molecule is formed, and dephosphorylation reaction is carried out by this enzyme, sucrose 6-phosphate phosphatase. Now as you can see, this whole process is happening in the cytoplasm, and in cytoplasm the sucrose molecules are being formed, and in addition to this inorganic phosphate is also formed. So as I told you earlier, the transport of triose phosphate molecules, which are precursors for the sucrose biosynthesis, happens from the chloroplast to the cytoplasm because of these inorganic phosphate molecules. They help in transporting, or you can say translocating, the sucrose molecule, triose phosphate molecules outside the chloroplast. So this is the major thing which happens during sucrose synthesis. So you can see here, this is a plant cell, and it also depicts how sucrose is loaded into the source tissues. Loading, once the sucrose is synthesized in the cytoplasm of the plant cells. So here, this is the sucrose molecule. Once it is synthesized in the cytoplasm, the concentration of this molecule builds up in cytoplasm. It has to be transported. It has to be transported to different parts of the plant. So that is called as loading. So these phloem vessels, they will be loaded with the sucrose molecule once it has been synthesized. So we'll see again. So this is the mesophyll cell, and in that mesophyll cell you have this chloroplast in which the Kelvin cycle or the C3 cycle runs. Photosynthesis happens. Carbon dioxide is fixed in presence of light, leading to formation of triose phosphate. Now, triose phosphate will be transported into the cytoplasm depending upon the concentration of this thing, inorganic phosphate. So if the inorganic phosphate concentration is high in cytoplasm, it will enter into chloroplast, and simultaneously this triose phosphate will move out. Because triose phosphate is important, because triose phosphate is not only involved in sucrose synthesis, as you can see here, but it also leads to formation of starch. So concentration of inorganic phosphate is important, as I'm telling you again and again, because if the concentration of inorganic phosphate is high in cytoplasm, sucrose synthesis will happen, because that will lead to simultaneous transport of triose phosphate out of chloroplast. But if inorganic phosphate molecules or concentration is low in cytoplasm, triose phosphate will be retained in chloroplast, and that will commence the process of starch biosynthesis. So this is a very important thing, and it has a very important role to play in plant development. So basically what you have to see here is that the triose phosphate molecules which come out from the chloroplast, they get converted into fructose 6-phosphate, and this fructose 6-phosphate, it combines with this UDP glucose, as I told you, that uridine diphosphate glucose molecule, and in presence of this sucrose 6 phosphate synthase enzyme, it gets converted into sucrose phosphate, and then this sucrose phosphate is dephosphorylated using this sucrose 6 phosphate phosphatase enzyme, leading to release of inorganic phosphate and sucrose. Now, another molecule which is formed during this process is trehalose. This trehalose molecule T. Yeah. So this T is trehalose. So some amount of this UDP glucose, it also combines with glucose 6-phosphate, and this in presence of this enzyme trehalose phosphate synthase results in formation of this trehalose 6-phosphate, and this trehalose 6-phosphate, again the phosphate molecules will be released, inorganic phosphate molecules will be released, and this will happen in presence of this enzyme trehalose 6-phosphate phosphatase enzyme. So here also you can see this enzyme is also phosphatase, this enzyme is also phosphatase. This is leading to release of phosphate group from sucrose, this is leading to release of phosphate group from trehalose. This trehalose is ultimately formed. Now trehalose again is important. It is an important signaling molecule in plants, and if it is in enough concentration, it can also get converted into glucose molecules, 2 glucose molecules. So this is what happens during the loading process of the sucrose in the source tissues. High concentration of sugar gets built up because of biosynthesis reaction or biosynthesis steps of this molecule, and once it is built up in the concentration is built up in the cytoplasm, it will transfer into the phloem tissues, which are made up of these sieve elements and companion cells, and once the sucrose molecule enter into the phloem cells, they will be ready for getting transported into various parts of the plant, and this transportation of sucrose molecules into the phloem cells happen again through two different you can say paths. One is through the plasmodesmata, and the second one is through the integral proteins. These are the sucrose transporters, you can say sucrose transporters, which allow the entry of sucrose into the sieve elements and the companion cells of the phloem tissue. Now, once this sucrose molecules are loaded into the phloem tissue near this source tissues, which is the leaf, which is producing the sucrose, what happens is you can see sucrose is a crystalline solid, and once it is dissolved in water, it becomes it acts as an osmolyte, so it creates a turgor pressure in the phloem tissues, and this has an osmotic effect, and this difference in the turgor pressure drives the mass flow of water carrying the assimilates. These assimilates means these sucrose molecules and other nutrients, of course, to the sink tissues where the sucrose synthesis is not happening. So the sucrose controlled phloem transport is the predominant pathway through which all resources, including water, nutrients, signaling molecules, for example, like this trehalose, are imported to the meristematic sinks or the meristematic tissues, including the shoot and the root meristems or apical meristems, the growing tissues, the growing shoots, the growing roots, where it is required for carrying out respiration. so that the energy can be released and the tissues can grow. So at a cellular level, sucrose is the key carbon source for growth and development and also defense of the plant cells. So sucrose metabolism, once it enters the catabolic pathway, it leads to formation of hexoses, the fructose and glucose, which are the subunits which make this molecule, and which make these molecules, and they are further utilized in respiration to release energy, or they can again be used by means anabolic processes leading to formation of several other polysaccharides like cellulose, starch, fructan, proteins, and antioxidant compounds. So since sucrose is made up of this glucose and fructose subunits, so once it reaches the tissues which are requiring sucrose as the source of energy, it will be broken down, converted into six carbon molecules of this glucose and fructose subunits, utilized in respiration, or it may again be used to build up during anabolic reactions to form starch, cellulose, etc. Now coming on to the unloading part of sucrose unloading in the sink tissues. So what you'll see here is once the sucrose reaches the tissues which require it as a source of energy. So these tissues are the tissues which are not able to synthesize organic carbon because they are non-photosynthetic tissues, and in non-photosynthetic tissues what will happen is that sucrose which has reached here, it will be unloaded uh, into these tissues, and they will enter into these cells through various mechanisms. And first of all, like one mechanism is that sucrose is broken down by the cell wall invertase enzyme, which is associated with cell wall, the CWIN, cell wall invertase. It is broken down into glucose and fructose. Then this glucose and fructose enters easily into the plasma membrane and then into the cytoplasm, and this can be received by receptors, these RGS receptors, regulator G protein signaling receptors, which are further activated, which lead to gene expression in the nucleus, and that leads to growth. The cell cycle commences, or you can say the reproductive or vegetative growth happens because of this. Another way by which the sucrose is taken up into the you can say the sink tissues is through the plasmodesmata. So sucrose coming through this phloem will enter directly through the plasmodesmata, and and in this it will reach the it will reach the cytoplasm, where it will be again broken down into glucose and fructose, which can be further utilized by glycolysis to derive energy. And this glucose and fructose can also act as precursors, or you can say activators of this hexokinase HXK, this hexokinase enzyme, which also has an impact on gene expression, leading to growth and development of the tissues. Then another way by which the sucrose can enter into the sink tissues is through the sucrose transporters. These sucrose transporters, which allow the entry of sucrose molecules and once the sucrose enters the cytoplasm, it is broken down into glucose and fructose using this cytoplasmic invertase. The two types of invertase are there, you can see, one is this cytoplasmic invertase and the other one is this cell wall associated invertase. So cell wall associated invertase will break down the sucrose molecule outside the cell into glucose and fructose. They will enter, they will have an impact, means they will activate these RGS receptors, which will further activate the gene expression process, which is required for activating cell cycles, cell growth, etc. And in same way, sucrose, when it enters from the phloem tissue through these sucrose porters or transporters, the cytoplasmic inverters, they convert this sucrose into glucose and fructose. And then the third mechanism is the entry of sucrose through plasmodesmata. Now, when we say that these three mechanisms, so they can be divided into this apoplastic and symplastic pathway. Now, mostly the sucrose means transport in all the plant cells or tissues, they happen by these two pathways. So you can see the difference between this apoplastic and symplastic pathway. So these are two adjacent cells, two adjacent plant cells. So these red zone, or you can say the red arrow, this indicates the apoplastic pathway. So it involves transport of materials without touching the cytoplasm. Okay, so because it is happening through the cell wall and it is not involving cytoplasm, this pathway is very quick. The transport through this pathway is very quick, but whereas the simplest pathway or the symplastic pathway, that involves the movement of molecules through cytoplasm between the cells to adjacent cells, you can say intercellular transport, and this happens through plasmodesmata. Now, if you compare the speed of apoplastic and symplastic pathway, you'll find that the molecules move slower in the symplastic pathway. Now, it is very easy to understand because if you have the analogy of movement of a person in, suppose, on a road, and movement of a person in water. So obviously in water you have to like exert more energy and your movement will be slow as compared to your movement on road. So that is the best analogy to explain the speed of the apoplastic and symplastic pathway. Now what we basically have to understand is that sucrose molecules also, they also move between. So this sucrose which is entering through this plasmodesmata, this can be through adjacent cell also. And another part, two other means you can say methods by which sucrose enters into the cell will be either through this sucrose transporters or through the cell wall by the action of these cell wall invertase enzymes, which will break down sucrose into glucose and fructose, and then glucose and fructose will enter into the plasma membrane, which will be permeable for glucose and fructose molecules. Because if you see the size of glucose, fructose will be of smaller size. They can easily enter into the plasma membrane, whereas sucrose molecule will be of larger size. It won't find easy pathway or easy way inside the plasma membrane. It will need special entry points which are made up of integral proteins, which is here you can see the sucrose transporters. So what you have to see is when sucrose reaches the non-photosynthetic tissues, which are called as sink tissues, because they do not synthesize organic carbon. They don't have the ability to perform photosynthesis. So sucrose will reach to the sink tissues through the phloem, and they will be unloaded into these tissues through these three methods. One is through plasmodesmata, this will be the symplastic pathway, and two apoplastic methods, apoplastic pathways. One is through the sucrose transporters, and another one is by breaking down of sucrose molecules outside the cell using cell wall invertase. I hope you have understood this point, so we can go on to the next topic. Now in the next we can see what is the significance of sucrose biosynthesis. So apart from the major transported molecule, sucrose has a very important role in plant growth and development. It is the central molecule central molecule related to the production of food, feed, fiber, and fuel. Therefore, it is pivotal, very important for modern agriculture, economies, and even energy sustainability, considering the thing that nowadays sucrose is even used for making bioethanol. So this is very important. So carbohydrates, you can see that originate from sucrose. So it may be starch, it may be cellulose, or it may be fructans, many other molecules, carbohydrate molecules which arise from sucrose. They constitute about 90% of the plant biomass. So this makes sucrose crucial in determining the final yield of the plants or the crops. It's very important to us as agriculture scientists, the yield of the crop. So sucrose is very crucial in determining the yield of the crop. So crop production, as you know, it needs to be increased substantially. We can say it needs to be doubled in next 50 years to meet the demands of increasing world population. And this has to be achieved despite adverse impacts on the agriculture because of global warming, abiotic stresses, biotic stresses. So, despite all these drawbacks, or you can say problems, we need to increase our agriculture production. So, sucrose metabolism can be a major focus of research effects or research of efforts which can be undertaken to achieve these targets. And in addition to ensuring our food security, sucrose can also play a very important role in ensuring energy security for us because it can be converted into ethanol, which can be used as biofuel, uh, non-conventional source of biofuel. It can be easily recycled. You don't have to depend upon the limited supply of petroleum products. So sucrose metabolism is a key pathway which needs to be judiciously researched and utilized to increase not only the crop yield, not only to ensure food security, but it also needs to be undertaken with a great enthusiasm to ensure energy security also. So one more thing which is important here is the role of abscisic acid in modulating the abiotic stress with the help of sucrose. So it has been observed that even before the abscisic acid comes to play its role in the process of abiotic stress, sucrose translocation can be reduced. And it has also been observed that after the abiotic stress situations are experienced by the plant, resulting in increased secretion of abscisic acid, the abscisic acid also results in suppression of the activities of these cell wall invertase enzymes and the sucrose 6 phosphate synthase enzyme, which ultimately result in lowering of the glucose pool, which has all these cascading effects as we discussed just now. Now coming on to the next role of sucrose which it plays in the plants is it helps in establishing, it has a major role in establishing the mutualistic relationship, mutualistic symbiotic relationship between some of the plant species and fungi. As we know, this symbiotic relationship is very helpful to both these organisms, the plant and the fungi, The plant receives the nutrients such as phosphates, particularly phosphates, because of these fungi, and fungi in return get the energy, the food source from the plants, which is particularly sucrose. So sucrose hydrolysis and hexose transports are very critical for these fungi, which are called as arbuscular mycorrhizal fungi, and to form symbiotic relationship with the roots, in which the roots provide sugar to the fungi to support its life, and in turn the plant receives phosphorus and other nutrients from the fungi. Now, if you see the mechanism of this sucrose modulating the mutualistic symbiotic relationship of fungi and plants, you will see that fungi induce cell wall invertase. This is, so here you can see this is the plant cell, and this is the fungal cell, which is targeting the plant cell for the mutualistic symbiotic relationship, and there is a crosstalk between the plant cell and the fungi cell. So what happens is the vascular mycorrhizal fungi, they induce the uh, cell wall invertase. The cell wall invertase from the root cortex, which leads to hydrolysis of sucrose. The sucrose, which is released outside the root cells in the root soil interface, and this sucrose is broken down with the help of these cell wall invertase, which have been induced by the fungal cells, into glucose and fructose. And once they are in form of glucose and fructose, these are small molecules, they can easily get transported into the fungal cell through hexose carriers, and which are located in the fungal membrane, and may means they enter into the fungal cell, where they will be used to generate energy. In addition, the arbuscular mycorrhizal fungi may also induce the expression of cytoplasmic invertase. So this cytoplasmic invertase, here you can see the cytoplasmic invertase or the vacuolar invertase. So that will result in the host to generate hexoses, which will be produced inside the cytoplasm of the root cell, and these hexoses will then be transported through the apoplastic pathway. Just before, I think 2-3 slides, we discussed the apoplastic pathway, which happens through the cell wall, and it is very quick. So through that apoplastic pathway, the glucose and fructose will reach the hexose carriers, which are located in the fungal cells, and they will get entry into the fungal cells, where they can be utilized to generate energy by glycolysis and other respiratory processes. So in return, the plant gets the phosphates, phosphorus in return. So what happens is the fungi, they take up the orthophosphate from the soil using high affinity inorganic phosphate carriers, which results in transportation of these inorganic phosphates into the fungal cells, and through unknown mechanisms they are shared to the apoplastic pathway, which helps in transporting the inorganic phosphate into the carriers present in the cortex, the cortical cells of the plants, which have been colonized by these fungi, and this results in transportation of the fungi into the root cells. That's how you have this mutualistic symbiotic relationship, in which you can see that the compounds which are produced by fungal cells, they lead to induction of enzymes like the cell wall invertase or cytoplasmic invertase or vacuolar invertase, and the function of all these induction processes or the inducing of these enzymes is ultimately breaking down the sucrose disaccharide. It's a disaccharide into glucose and fructose, which is then transported either through the apoplastic pathway into the hexose carrier present in the fungal cell, or sucrose may be transported directly to the apoplastic pathway, and there it will be broken down by the cell wall invertase into glucose and fructose and reach the fungal cells so that it can derive energy. And in return, the fungal cells will absorb the inorganic phosphate from the soil through this highly efficient inorganic phosphate carriers present in its membrane, and they will deliver this inorganic phosphate into this apoplastic pathway, this white region, the apoplastic pathway, as I told you earlier. This is the cell wall region between the cells, in which the movement of molecules is very quick, and these inorganic phosphates then get absorbed into the root cells, and that's how the phosphorus is absorbed by the roots in the plants. So this is another role of sucrose. It establishes, or it helps in establishing a mutualistic symbiotic relationship with the fungi in many plant species. Then coming on to another role of sucrose. So here you can see that sucrose is very important, and it plays a very critical role in alleviating the impact of pathogens, attack of pathogens on the plants. So plant pathogens like fungi, bacteria, viruses, they attack the plant to suppress the host immunity, and they obtain their nutrients mainly from sugars, which is basically the sucrose, for growth without returning any benefit. So this is not a beneficial relationship, and pathogens lead to destruction of the plant tissues and ultimately reduction in the crop production. So in response, many plants have developed ways by which they limit the availability of these nutrients to the pathogens. So this is a counterattack from the plant side, a defense activity or a defense mechanism by the plant which it employs to control the growth of pathogens. And this is one of the way where they limit the supply of nutrients and even sucrose also. So both processes are tightly influenced, you can see, by sucrose transport metabolism and signaling. And during this tug of war between the pathogens and plants, sucrose plays a very important role, and understanding this mechanism is critical for developing new solutions which can be used for controlling diseases. So what basically happens is pathogens try to hijack the whole cells. So this is here, and you can see this is the plant cell. This is the plant cell in which we have this nucleus, and we have the cell wall invertases. These are the you can say porters, you can say sweet effluxers, hexose carriers. These porters are there, and these are the pathogen cells. So basically what happens is the pathogen cells, they try to hijack the host cells of the plant to get the sugar molecules. They activate the sweet receptors, expression of these sweet receptors, and the cell wall invertase, which lead to conversion of sucrose into glucose and fructose. And this is the major strategy employed by the pathogenic bacteria. And these effector proteins secreted by bacteria, they bind and activate the promoter of sweet sucrose effluxer, the sweet sucrose effluxer. And it is likely that the cell wall invertases are also activated by these effector molecules. And sucrose is hydrolyzed into glucose and fructose, which are then taken up by the bacterial hexose carriers. And in response, the plant cell retrieves the hexose through a hexose carrier to starve the pathogens. So you can see there is a competition between the hexose carrier of the bacterial cell, or you can say the pathogen cell, this red one you can see here, and the hexose carrier of the plant cell. There is a tug of war, you can say, between these two cells, and they compete for the glucose and fructose which have been formed because of the sucrose molecule being broken down due to the activation of the cell wall invertases and transport of sucrose outside the plant cell through these sweet effluxer proteins which are present in the plasma membrane of the plant cell. So as a countermeasure, bacteria produce molecules, decoy molecules, or you can say molecules which fool the plant cells. So the one of the molecule which is used as a countermeasure by bacteria is trehalose, this pink color molecules. These pink color molecules are decoy molecules to fool the plant sensing. The hexose carriers of the plant, they absorb the trehalose instead of the hexoses like glucose and fructose, which are required for energy producing reactions. So bacterial cells will result in formation of these molecules trehalose, which disrupt the host sensing and the defense mechanism, and they achieve their target of getting energy from glucose and fructose, and that's how the pathogen establishes its supremacy against the plant cells in due course of time if good care is not taken after the infection. So we have seen now how sucrose is very important for and plays an important function to perform. It has a very diverse functions to perform in the plants, and so it is very means once we have studied the biosynthesis part of sucrose, it is important to know how the regulation of sucrose biosynthesis happens in plant. So as we have seen earlier, sucrose 6-phosphate synthase, this is the enzyme which is principally involved in synthesis of sucrose. And so if anything which controls this sucrose 6-phosphate synthase enzyme is an important tool to regulate sucrose biosynthesis. It has been seen that the activity of these enzymes, the sucrose 6-phosphate synthase enzyme, is greatly affected by phosphorylation process. Phosphorylation means addition of a phosphate group to one of the amino acid residues of the enzyme. Enzyme is after all a protein, so it will have amino acid residues, and you'll see that one of these amino acid residues, if they are phosphorylated, it leads to less activity of the enzyme, and if they are dephosphorylated, they will lead to more activity of the enzyme. And this phosphorylation process is carried out by an enzyme called as sucrose 6-phosphate synthase kinase, SPS kinase enzyme. So any molecule which modulates the action of this SPS kinase enzyme will have the impact on sucrose 6-phosphate synthase. So you can see this molecule, glucose 6-phosphate. So glucose 6-phosphate is it directly stimulates this SPS kinase. It inhibits SPS kinase by activating sucrose 6-phosphate synthase. And in dark conditions, this phosphate directly inhibits. So you have in dark conditions, what will happen? There will be less availability of glucose 6-phosphate. There will be high availability of inorganic phosphate. So this inorganic phosphate will inhibit the SPS phosphatase, thereby inhibiting the Sucrose 6-phosphate synthase enzyme. So basically what you have to remember is that the regulation of sucrose biosynthesis is mostly done through the sucrose 6-phosphate synthase enzyme, and one of the mechanisms to control this enzyme is by controlling the SPS kinase, phosphorylation or dephosphorylation of the enzyme. This linear chain is there, and this linear chain is formed by attaching of the glucose subunits by a alpha 1,4 glycosidic bond. So this is the first carbon of the glucose molecule. This is the fourth carbon. You can see 1, 2, 3, 4. This is the first. So that's why it's called as alpha 1,4 linkage. And this alpha 1,4 linkage leads to creation of this linear molecule, which is called as amylose. And in addition to this amylose, starch also have branched molecules which are formed when we have alpha 1,6 linkage. You can see here, it is here. So this is the sixth carbon atom, and this is the first carbon atom. So this 1,6 alpha 1,6 linkage leads to branching of the molecule, which results in formation of amylopectin. So amylose and amylopectin are the two components which make the starch molecule. And some other features which are important regarding starch is like starch is insoluble polymer, obviously, because it's a large molecule. And even branched molecules, it has branched components also in form of amylopectins. So it is insoluble in water. And second thing is it is synthesized inside the plastids. Now if you compare the synthesis of sucrose and starch, this is the major difference. Mostly sucrose, you will see that they are synthesized in cytoplasm, whereas starch is synthesized in the plastids, mainly chloroplast of higher plants. And another feature is about starch, and with the differentiating feature you can say of sucrose and starch is that sucrose is the major transportable carbohydrate in plants, whereas starch is the major storage carbohydrate. That's another difference. And it has high economical importance because it is the second most abundant biopolymer on earth after cellulose. Uh, Another importance of starch is as food and feed purposes, most important carbohydrate in terms of food and feed. Everyone is taking potato in food, and that's high amount of starch. It has high amount of starch, and many cereals also they have high amount of starch. Similarly, feeds which are provided to animals, they also have significant amount of starch. And in addition to that, starch also has industrial value, particularly for bioethanol production. And you see the steps of starch biosynthesis here. So the first thing is that the synthesis process will happen in chloroplast. You can see here, this is suppose a leaf cell, having a plant cell of a leaf. So this is the cytoplasm part where sucrose synthesis will happen, and this is the chloroplast part where the starch synthesis happens. And the first committed step during starch biosynthesis is conversion of this glucose 1-phosphate. You can see here this glucose 1-phosphate. This glucose 1-phosphate is converted into ADP glucose with the help of one ATP molecule. So basically what happens is this ATP releases 1-phosphate, inorganic phosphate molecule, and it results in formation of ADP, and this glucose combines with this ADP to form ADP glucose, and 2 phosphate molecules are released, and this process is catalyzed by this enzyme ADP glucose pyrophosphorylase, or it is also called as AGPase. Okay, AGPase enzyme. Now ADP glucose acts as the glucosyl donor. It will act as the glucosyl donor for different classes of starch synthesis, There are many types of starch synthesis present in plants, which elongate the alpha-1,4 linkages in the glucan chain of the two insoluble starch polymers, the amylose and amylopectin. If you remember, this linear chain was amylose. The branched chain having the alpha-1,4 and the alpha-1,6 linkage was amylopectin. So this ADP glucose acts as the glucose. It will provide the glucose residues for elongation of that chain, either in the linear form and also in the branched form, leading to formation of the starch granule. Now you'll see that in leaves, during light or in abundance of light, glucose 1-phosphate is synthesized from the Kelvin cycle intermediate via plastic phosphoglucose isomerase and phosphoglucomutase, while ATP is provided by phosphorylation at the thylakoid membrane. So here you can see this Kelvin cycle will result in formation of triose phosphate molecules, and these triose phosphate molecules can combine and result in formation of glucose 6-phosphate. So this glucose 6-phosphate is then converted into glucose 1-phosphate. This is just a mutation reaction which is catalyzed by this phosphoglucoisomerase and phosphoglucomutase enzymes. And then the enzyme is the principal enzyme of starch biosynthesis, that AGPase enzyme comes into action, resulting in formation of ADP glucose. And this ADP glucose serves as the molecule which provides the glucosal residues for building up the amylose chain and the amylopectin chain of the starch. So this is about the starch biosynthesis, and these are the enzymes which are involved in this reaction. So you can see here one here. So this is the first enzyme, phosphoglucoisomerase, in which this glucose 1,6-phosphate is formed. Then we have the second enzyme is this one, phosphoglucomutase, in which glucose 6-phosphate is converted into glucose 1-phosphate. Then third enzyme is AGPase, which results in formation of ADP glucose. The fourth enzyme is starch synthase. Then you have the starch branching enzymes, starch debranching enzymes from 4th to 6th step. And 7th is this inorganic phosphate, which is released in the plastid. So you can see the buildup of the inorganic phosphate molecules is inside the chloroplast. Now if you remember the sucrose biosynthesis, what we noticed was that when inorganic phosphate concentration is high in cytoplasm, in that case triose phosphate molecules come out and it is utilized for sucrose synthesis. But if it is high in chloroplast, you'll see that the triose phosphate translocation outside the chloroplast will be lowered. It will be reduced, and they will be utilized then for starch biosynthesis. That is the important role of inorganic phosphate. Concentration of inorganic phosphate in the cytoplasm of the plant cell has a great impact on deciding whether sucrose synthesis will happen or starch synthesis will happen in the plant cells. Yeah, so this glucose 6-phosphate will be transported into the amyloplast through this glucose 6-phosphate translocator here present in the amyloplast membrane, and it is converted again into glucose 1-phosphate. Then it is converted into ADP glucose utilizing the ATP molecule here, and then it results by same steps which we have seen previously in the leaf cells into starch. So this ADP glucose provides the glucose residues leading to formation of amylose and amylopectin and ultimately starch. Now one small modification is observed in cereals. So you'll see this pathway of sucrose getting converted into UDP glucose, fructose, then UDP glucose getting converted into glucose 1-phosphate, glucose 6-phosphate entering into the amyloplast membrane through this glucose 6-phosphate translocator in potato tubers. These chain of events happen in potato tubers, but in case of cereals, what it is seen is that UDP glucose is converted into glucose 1-phosphate, and in outside the amyloplast itself, in the cytoplasm itself, ATP is used to form ADP glucose from glucose 1-phosphate, releasing phosphate, inorganic phosphates inside the cytoplasm itself. And this ADP glucose then enters the amyloplast through this ADP glucose translocator. Here it is, which is present in ADP glucose translocator. It's integral protein basically, through which the ADP glucose molecule enters directly into the amyloplast, and then it results in formation of starch. So this is the difference. So in potato tubers, the pathway will have these chain of events, and the entry will be of the glucose 6 molecules through the glucose 6-phosphate translocators. Whereas in cereals, you will see that the glucose 1-phosphate will not get converted into glucose 6-phosphate. It will get converted into ADP glucose, and ADP glucose will enter through the ADP glucose translocator, which will ultimately result in formation of starch. So that is the difference between the potato tubers, synthesis of starch in potato tubers, and cereal cells. I hope you have understood this. So next is you can see the steps of starch synthesis, the growing ends of starch synthesis here in various steps. So you can see this is the ADP glucose, which provides the glucose residues for increasing the chain length. So suppose this is the first glucose molecule which is provided by ADP glucose, then the second molecule comes, it attaches to the first molecule, then we have, in this way, the growing chain is there. Subsequently one more ADP glucose will come, it will provide the glucose residue on the reducing end. It will be, the end will be the reducing end, and ultimately the step will be followed for a long period of you can say subunits for enough time so that large number of subunits get attached in linear as well as branched form, leading to formation of starch ultimately. Now coming on to the process of regulation, it's very important. So regulation of starch biosynthesis, you can see so ADP glucose phosphorylase is the principal enzyme involved in the regulation of starch biosynthesis. ADP glucose pyrophosphorylase is the principal enzyme required for synthesis of starch. That's why it is the principal enzyme on which the regulatory activities will have their effects. So it has been observed that this enzyme is inhibited by high concentration of phosphates, which are accumulated in the dark in the cytoplasm. You can see that's what we have seen. High concentration of inorganic phosphate in cytoplasm results in sucrose, enhanced sucrose biosynthesis, and it is activated by high concentration of 3-phosphoglyceric acid inside the chloroplast. So these are the two things which have their impact on the ADP glucose pyrophosphorylase enzyme. So it is inhibited by high phosphorylase inorganic phosphates, and it is activated by 3-phosphoglyceric acid. So when inorganic phosphate levels are high in cytoplasm, triose phosphates produced in Kelvin cycle are exchanged, and this leads to promotion of sucrose synthesis and inhibition of starch synthesis. Whereas if the inorganic phosphate concentration is low in cytoplasm, triose phosphate is retained in the chloroplast and it is used for starch synthesis. So that's what I was telling you continuously, that the concentration of inorganic phosphate level is a key factor in deciding whether the plant cells will synthesize sucrose or they will synthesize starch. So this is the comparison of the, you can say, summary of the starch and sucrose biosynthesis, and you can compare the two things very easily here. So you can see this starch biosynthesis is the principal storage carbohydrate, whereas sucrose is the principal transporting carbohydrate or transport form of carbohydrate. Then starch biosynthesis occurs in plastids. It may be chloroplast, it may be a myeloplast, whereas sucrose biosynthesis will happen in cytoplasm of the plant cell. Now the precursor you can see is here ADP glucose, whereas in sucrose the precursor is UDP glucose and fructose 6 phosphate. In starch you can see that the addition of the monomers happens on the reducing end, whereas in sucrose also addition happens by formation of a glycosidic bond. And if you see the nature of glycosidic bonds, in case of starch the bonds are alpha 1,4 glycosidic and alpha 1,6 glycosidic bonds. Alpha 1,4 glycosidic bonds result in formation of a linear chain, which is amylose, and if there is a branching because of alpha 1,6 chain, it will result in formation of amylopectin. Whereas in case of sucrose, the nature of the glycosidic bond is alpha 1 beta 2 linkage, and these are the two subunits. So I think we have enough discussion on the starch and sucrose biosynthesis, and these two papers I have used to prepare the presentation for this topic. These papers are very informative and available on Google Scholar as open access articles. You can consult these articles plus additional resources like textbooks for biochemistry and online resources so as to prepare your notes and further improve your understanding. And I hope you may have understood the topic, and in case of any doubts, feel free to get in contact with me anytime. Thanks and good luck.